important in the context of relapse, but doesn't seem to be playing a direct role in the pathogenesis of the disease. It's not being positively selected in the absence of chemotherapy. Moreover, when we actually looked at what is NT5C2, we actually found a direct link on the action of the protein encoded with this gene with activity of chemotherapy. NT5C2 is what we call a cytosolic nucleotidase. It's part of the machinery that builds the building blocks that synthesize DNA. DNA is absolutely essential for cell viability, growth, and proliferation. And increased synthesis of DNA is a major characteristic for leukemia cells to grow and proliferate. The way we treat leukemia is in well part by actually blocking DNA synthesis. And two of the drugs that we use to block DNA synthesis and actually damage the DNA and induce activation of cell death are nucleoside analogs. They are poisons that actually enter the cell and enter the DNA synthesis machinery, inducing toxic metabolites that will block DNA synthesis and actually damage the DNA. These are 6-mercaptopurine and 6-thiogonine. These are the key drugs in maintenance therapy. This is the drug that the patients take every day for 2 years, once they are fine and remission, trying to control the progression of relapse. So what NT5C2 does is once these 6-mercaptopurine and 6-thiogonine are metabolized in the cell and incorporated in the DNA synthesis, it can actually inactivate the effectors of these drugs by dephosphorilating thiamp, thiaxmp, and TGMP, which are the active toxic metabolites. Removing this phosphate allows the cell now to export this out. So we have a mutation in the protein that is in a key pathway for leukemia growth and a key pathway for the action of chemotherapy. However, this is exactly the opposite that would happen. Why would we have mutations in the gene? They think antagonizing the effects of chemotherapy. You would expect that you have mutations in genes that are actually promoting the action of chemotherapy, that would antagonize the action of chemotherapy. So with this paradox, we looked at more patients and we sequenced a very broad spectrum of leukemia. And when we picture the whole spectrum of NT5C2 mutations that we saw, we find another pattern. And the second pattern that we find is not only these mutations are recurrent in multiple patients, they are recurrent in the positions that they affect in the structure of DNA and in the proteins that are made. And that what this tells us immediately is that these are not loss of function mutations. There are multiple different ways in which you can actually kill the function of the gene, but if you want actually to re-engineer the function of a protein and make it work better, there are only so many knobs that you can actually do. And you can see how many of these mutations are recurrent in position and also recurrent in the kind of amino acid substitution that you'll find, suggesting that this would actually be gain-of-function mutations, making the enzyme better, and in this way allowing it to antagonize the effects of chemotherapy better. So to prove this hypothesis, we generated recombinant protein and we tested its enzymatic activity, and in fact, each and every mutation that we find in a relapsed patient has gain of enzymatic activity. This not only has increased enzymatic activity in response to ATP, which is the physiologic activator of the protein, but also cells actually become ATP independent. NT5C2 is inactive. It's an inactive enzyme that requires to be activated by ATP. What these mutations are doing is locking the protein in the activated state. Now the switch on mechanism is constitutively active. This enzyme is going to be working all the time, allowing now the cells to escape the effect of chemotherapy. And we know this happens at the molecular level because we know how NT5C2 is activated. There is a molecular switch that's represented here. A region of the protein is in a disorder and structure and configuration. When ATP, the physiologic activator, binds, it adopts an helical configuration, and this helical configuration allows the substrate, the physiologic substrate in pink, to actually be catalyzed and be metabolized. When we looked at what are the effects of the mutations that we find in these patients, what we see is that this region that is in the normal protein and structure waiting for the ATP activators to become an alpha helix, it's always in an alpha helix configuration. This mutation, lysine 359 to glutamine, actually induces water exclusion of this region, locking this switch in the own alpha helix configuration and resulting in constitutive activation of the enzyme. The prediction that we will make out of this is that first, this mutation should confer resistance to chemotherapy, and they should confer resistance specifically to 6-mercaptopurine chemotherapy, and this is the case. When we express these mutations in cells, and we treat with increased doses of 6-mercaptopurine. You can see in the red line we can effectively clear leukemia cells. The blue line represents the effect of the wild-type protein, which is not very active in conferring resistance, but you can see in the line in black how the expression of this leukemia relapse-associated mutants results in almost 3 logs of differential response to chemotherapy. Massive progression of that we would predict would result in massive progression on the therapy. However, if you want to actually define progression under therapy, you cannot do this in a petri dish. You need to do it in a patient or in an animal model. And to test this, we generated a conditional inducible knock-in of this mutation, so we can actually introduce the mutation specifically and in an inducible way in the NT5C2 locus. In the mouse, we generate leukemias out of these mice, and these are leukemias that we can switch from a wild-type configuration to a mutant configuration, and now test the effects of chemotherapy. And what we see is that the system works very effectively. We can specifically activate the mutation. As you see in the line of red, these cells that now has differential activity, just the presence of this mutation, are 3 logs more resistant to treatment with 6-mercaptopurine in vivo than the wild-type cells. And the second prediction that we would make is that when we look at these patients that relapse with NT5C2 mutation, we predicted that these patients would actually relapse while receiving 6-mercaptopurine. They should probably relapse across escaping from the effect of the therapy. And when we look at the pattern of mutations, what we see is that the NT5C2 mutations are associated with early relapse. Each and every patient that has an NT5C2 mutation relapse while taking 6-mercaptopurine, while receiving this maintenance therapy over 2 years that was aimed at preventing the occurrence of relapse. Now linking directly the pressure of this very specific drug with the occurrence of this very specific mutation. And these are the questions that we have on top of the table. So first, are we still missing driver mutations? And we think we are still missing driver mutations. Exome sequencing covers 5%, in best cases 7%, with high coverage of the leukemia genome. There are probably mutations in the non-coding space that are contributing to relapse. We're very interested in knowing what is happening there. Our 18 most common driver mutations account for about 60% of the mutations of the patients that relapse. We still have to find what's happening in the other 40%, and we are sequencing deeper and farther now to find this. We want to understand better what are the evolutionary dynamics, what really matters in this process of clonal selection. Is that the mutation rate and the spectrum of diversity matter, or is actually the occurrence of very specific mutations what ultimately will result in the selection of a winner clone that will escape the effects of chemotherapy? And finally, I'm focusing on NT5C2. Can we actually now design a mutant-specific inhibitor that would allow us now to treat these patients, return these leukemia cells to a state of sensitivity to chemotherapy, either to treat the relapses or perhaps even more importantly, to prevent the occurrence of these mutations, and combination with 6-mercaptopurine will actually make this drug even more effective, cutting the escape mechanism during maintenance therapy, and in this way, cut down the way to relapse. I have to acknowledge the people that have been instrumental in this work. Much of the data presented today here is the work of Dennis Makeba, Chelsea, and Richard Shima in the lab, and nothing of this would have been possible with the help of Raul and Teresa in the sequencing and in the data analysis. And with this, I'll thank you for your attention. I'll take any questions.
Thank you. This is fascinating. You are talking about probably, and I'm going to the last slide that you have here. Vamos a cambiar al español. En la última diapositiva que había, en la que presentaba la opción de utilizar inhibidores de este NT5C2. Sin embargo, es un gen que no está mutado inicialmente en el proceso de estas leucemias linfocíticas crónicas. ¿Cómo? Y hoy estamos moviéndonos a un campo de medicina preventiva, en donde lo que buscamos son marcadores de genes que nos sirvan para poder predecir si un paciente tiene un determinado panel indicativo de un diagnóstico. ¿Cómo? No entiendo, o sea, si este es un gen que es el resultado de este tratamiento con el mercaptopurine, y aparece posteriormente, ¿cómo es posible detectarlo? ¿O cómo vas a conseguir un inhibidor que utilice simultáneamente con el mercaptopurine en el comienzo del tratamiento?
Sí, es muy buena pregunta. Hay dos versiones de cómo ocurren estas mutaciones. Hay grupos que consideran que estas mutaciones están presentes al diagnóstico o presentes al inicio del mantenimiento del tratamiento. Nosotros no las vemos. Si están, son clones muy minoritarios y hemos mirado muy profundo. Lo que nosotros pensamos es que esta mutación tiene un doble efecto. Es una mutación un poco paradójica. Esta mutación representa un perfect gain of fitness en el contexto del tratamiento con mercaptopurina, porque le permite a la célula eliminar esta droga fuera del compartimento celular y escapar del efecto del mantenimiento. Sin embargo, el efecto de la mutación tiene un precio. Y el precio que las células pagan es que al mismo tiempo que NT5C2, el mutante NT5C2 va a estar detoxificando a la célula y exportando los metabolitos tóxicos de la 6-mercaptopurina, también va a estar exportando los componentes fisiológicos de la síntesis de purinas. De manera que el precio que paga por escapar del efecto de la droga es disminuir su capacidad proliferativa. Y ahora sabemos también incluso disminuir sus actividades tensas. Lo que pensamos es que antes de introducir el tratamiento con mercaptopurina, esta mutación está contraseleccionada. Es una mutación que en el proceso de selección clonal darwiniano es perdedor. Los clones que tienen esta mutación van a ser eliminados por competición con otras células de la leucemia que no la tienen. En el momento en que introducimos la 6-mercaptopurina, cambiamos las reglas del juego. Y ahora estos clones, la ocurrencia de este clon muy minoritario o simplemente la ocurrencia de esta mutación en algunas de las células que están quizá en un nicho protector o de leucemia stem cells que están esperando la oportunidad de escapar del efecto de quimioterapia, es lo que le permite escapar. ¿Cómo se traduce esto en esquemas terapéuticos? Lo que nosotros pensamos es que NT5C2 es una diana perfecta. Y es una diana perfecta porque es inactiva. Las células no utilizan esta enzima. El enzima wild type, su función fisiológica es estar en un compartimento inactivo y en caso de que la célula necesite activarlo, activa este switch conformacional para exportar nucleótidos en un momento determinado. La droga debería no afectar masivamente la actividad celular. Si combinamos un inhibidor, incluso en la ausencia de mutación, nosotros no pensamos que haya que buscar el paciente que tiene la mutación para dar la droga. Nosotros lo que pensamos es que la droga habría que dársela a todos los pacientes. Todos los pacientes tienen la oportunidad de desarrollar un clon con una mutación que resulte en el escape al tratamiento. Si damos 6-mercaptopurina con el inhibidor de NT5C2, lo que vamos a estar es eliminar esa posibilidad. Ese winning ticket para la ocurrencia de recaída ahora está fuera del mapa de todos los pacientes, con lo cual las recaídas, en lugar de ser el 20%, deberían ser el 15% o el 10%. No lo sabremos hasta que no tengamos ensayos clínicos, pero ahí sí es como nosotros lo
Muchas gracias, Adolfo. Tenemos un último ponente que no está en el programa. Quería dar un anuncio y medio. El primero es que la sesión social va a ser en la ICC, no va a ser aquí. A continuación vamos a tener la última sesión de biomedicina del encuentro. Y el segundo es que nos encantaría hablar de medicina personalizada, de Big Data y todo eso que metemos va a ser en el café, ¿vale? Así que perdonadme porque el debate no se vaya a acabar. Nuestro último ponente es el doctor Carlos Cuchaga. Hizo su tesis doctoral en bioquímica y biología molecular en la Universidad de Navarra. Él empezó a estudiar las causas genéticas de las enfermedades neurodegenerativas durante su postdoc y completó su formación en neurogenética en esta universidad, en la Universidad de Washington University. Actualmente es profesor asociado del Departamento de Psiquiatría y Neurología en la Universidad de Washington, en San Luis, Missouri, y dirige el laboratorio de neuroinformática. Su investigación fundamentalmente está centrada en la identificación de genes implicados en enfermedades neurodegenerativas, siendo uno de los pioneros en el uso de biomarcadores y secuenciación para estudios genéticos de Alzheimer. Sí, por favor. Doctor Chaga.
Muchas gracias. Bueno, como veis, yo no voy a hablar de Cancer Genetics y también sé que vamos retrasados, así que no os preocupéis, mi charla va a ser bastante corta y va a ser muy general. Vamos a hablar de lo que sabemos de la enfermedad de la genética de Alzheimer y muy poco de lo que yo hago en la anterior. Entonces, vamos a empezar porque no tenemos tiempo. Okay, so I'm going to have 3 parts in my talk. We'll have a short introduction to know what we know about the biology of the disease, and then I will go through 2 different steps that are related to the genetics findings for Alzheimer's disease. We'll cover GWAS and also next generation sequencing. So why genetics is important? I think this is a key question, right? We think that the goal of my lab and many other labs is to understand the biology of the disease. And to do this, we use an unbiased approach, which is genomics. The idea is to identify novel genes, novel variants that are implicated in the disease. And doing this, we think we want to identify the major pathways, and by doing this, we think we understand the disease. If we understand the disease, we may be able to identify new therapeutic targets. And this is important because there is no therapeutic target for any genetic disease. I think this is key. Okay, so we go to the next one. And again, this is the same question, why genetics is important? Right now, most that we know about Alzheimer's disease start with genetics. If we understand the genetic basis of the diseases, we may be able to have better prediction tools. And also, by identifying the genes, we are able to identify new targets. And I will show some examples here today. Okay. Sorry. Go back. Okay, so Alzheimer's disease is the most common genetic disease, although there are several. We have frontotemporal dementia, CDD, PSP. Alzheimer's disease is the most common. It's characterized by dementia and changes in behavior and personality. Alzheimer's disease pathologically are characterized by the presence of 2 proteins in the brain. One is hyperphosphate tau, which is extracellular and forms tangles, and the other protein causes the plaques, and this is the protein that also against extracellular. So how common is Alzheimer's disease? Alzheimer's disease, right now there are more than 5 million of people only in America with the disease. It also causes around 100,000 deaths per year. And the major risk factor for Alzheimer's disease is age. You can see that the prevalence for Alzheimer's disease increased from 5 to 30 with age. Therefore, this is going to be a big problem, specifically for US, but also for European countries, because the amount of people in this age range in the next years is going to increase exponentially. Therefore, we expect that around 50 million of people in the US may be affected by Alzheimer's disease in 2020, although we don't have the numbers. Okay. Alzheimer's disease clinically can be classified into 2 classes. One is called sporadic Alzheimer's disease. This is the most common form of the disease. Is normally presents late onset, around 65, and is considered to be a complex trait. We know that there are genetic and environmental factors implicated in the disease. There is other form of disease which is called early onset of familial Alzheimer's disease. This form of disease normally aggregates within families and is considered autosomal dominant. Besides the age at onset, there is no difference in the disease in these 2 cases. Okay. So a lot of what we know about the Alzheimer's disease in general is because a long time ago, in 1991, several mutations in 3 genes, PCN1, PCN2, and EPP, were identified to cause Alzheimer's disease in very large families. And here you can see the first family in which the mutation in EPP was identified. And by studying the individuals in the family with the disease and individuals in the family without the disease, the mutation in APP was found in 1991. So the important thing is, as I said, that 3 genes, APP, PS1, and PS2. An interesting thing is that these 3 genes are part of the same pathways. You can see in this nice graph that we have APP in the middle, and APP is cleaves, and there are two potential pathways. One pathway that goes up is the non-amine pathway, and APP is cleaved by alpha secretases, right? Gamma secretases. And this creates a product which is soluble APP, which doesn't cause any problem. On the other hand, if we have the beta secretase pathway, we have another product which is again then cleaved by the gamma secretase, which generates this A-beta product, which is the protein that aggregates in the brain. As you can see, the gamma secretase is an important step in this pathway, and Precission 2 and Precission 1, they are both gamma secretase. Therefore, you can see that APP, PS1 and PS2 are part of the same pathways. And this is very important. Why? Because now that we know this pathway is very important for Alzheimer's disease in general, not only for the family Alzheimer's disease, we can start developing therapies for this disease. So we move to the next slide. This is the same graph, but now it's flipped. You have APP in the middle, and you have Precission 1 and Precission 2. Here you can see that a lot of, in the different parts of these pathways, we have potential therapies for Alzheimer's disease. And even the genes were identified in 1991, these therapies are now in clinical trial. So we have beta and gamma secretase inhibitors, and there is also a lot of interest these days in immunotherapy for A-beta and A-beta variants. So we are getting there. We are a little behind in the field compared with cancer, but now some of the findings that we found in the studies, they are moving to clinical trials. Okay, so as I say, most of these findings were based on studying the familiar form of the disease, but this is the rare form. So what do we know about the sporadic early onset of the disease? So, the major risk factor for late onset Alzheimer's disease is the ApoE4 allele. The ApoE4 allele increases the risk for Alzheimer's disease between 3 to 8-fold, depends on whether you have 1 or 2 copies of the A4 allele. As is very interesting, and the same gene, there is another variant, ApoE2, which is associated with decreased risk for Alzheimer's disease. So this is a protective variant, and we know about this for a while. ApoE, as you can see in the graph, is also associated with its onset, and again, this is the major risk factor for late onset Alzheimer's disease. ApoE is also involved in the APP pathway. Latest studies suggest that ApoE is associated with A-beta clearance and aggregation. So clearly, A-beta is a central pathway in Alzheimer's disease in both sporadic and late onset. So the question is, there are additional genetic risk factors for late onset Alzheimer's disease, and one way to study these factors is by doing genome-wide association studies. These are very common, especially in the last 10 years or so. In this study, what we do is we analyze millions of common variants in the genome. The latest studies normally between 6 to 8 million. And the most common study design is to compare the allele frequency of those variants in cases versus controls. But there are different approaches, also using family and using endophenotypes. In my lab, we have been using a lot quantitative biomarkers, especially in CSF, tau, and A-beta, and we have identified several genes implicated in Alzheimer's disease. I don't have today to talk about that. If you have any question, we need to talk about that. So you can see for these studies, we need a very large sample size, and this is the latest genome-wide association study for Alzheimer's disease that was published in 2013. In this study, 74,000 individuals were included. So this is an international collaboration with several groups in the US and several groups in Europe and UK and France, because in order to do the studies, you really need these results. This is the summary result of the study, and this is what is called a Manhattan plot, because it looks like Manhattan. And what we have here is each dot represents a genetic variant, and they are sorted in the X-Y axis by chromosome and restricted position. In the y-axis, what we have is the minus log p-value. Because we are testing so many variants, statistically we need to have a very strong p-value. So in order to be considered significant, in this study, we need to have a p-value in 10 to the minus 8, if you have the line there. As you can see, there are a lot of peak or signals that pass that threshold. And on top of those signals, we have a gene name. This is the gene we think is driven association, but we are really not sure because under those peaks, there may be more than one. The other problem of the GWAS is that effect size of those variants is relatively small compared with equal. One of those genes or signals may increase the risk for Alzheimer's disease, maybe only 2 or 3%. Besides all of these problems, if we move to the next one, the most important thing of these studies is that these genes, they are not independent between them. We see these genes are part of a specific pathways. Obviously, the ABRAD pathway is one of the most important pathways, but as you can see in this slide, immune response, synapse function, and others are also highlighted in these studies. And we think that, well, these genes has a very small effect size, but these genes are part of a specific pathway. If we are able to target that pathway, we may be able to identify treatment for disease. Okay, so this is all about common variants. There are more studies that are going on. Studies basically, as I said, with large sample size in non-final times. But the question is, well, how we can overcome the problem of the common variants? Well, let's see if we can identify low frequency and rare coding variants that have a large effect size. The good thing of these studies is also that you identify a specific gene that we can model in the lab, and we can do functional analysis in cell lines and animal models. So the first question is whether these variants exist and how we identify those. Obviously, we can do whole exome, whole genome, RNA-seq, and candidate genes. So in 2013, two different groups in which we part of one study, we identified a variant in another gene called TREM2. We have a large effect size for Alzheimer's disease. This variant was the R47A and was identified by doing whole exome and whole genome sequencing in different populations. One study was the Icelandic population, and in the other was a collaboration between US and UK. So interesting thing of this gene is that this gene has been always linked and studied because its role in immune response and inflammation, specifically with HIV viral infection. But nothing was known about this gene and Alzheimer's disease. So the role of these genes in the brain was totally unknown. Okay. So based on this and this, and also on the GWAS studies, clearly we think now that the immune response inflammation is a key event for Alzheimer's disease. As I said, Alzheimer's disease is characterized by the presence of plaques and tangles of the brain, but there are diseases that are also characterized by protein aggregation in the brain. So my lab and others, we also demonstrate that this gene is associated with FTD, frontotemporal dementia, Parkinson's disease, INLS. And the important thing is that we think that this gene is not affecting risk for the disease through an A-beta-dependent mechanism, but by affecting immune response and inflammation. Okay. So the question is, how can we identify additional rare variants in this disease? And here I want to remember that we are studying cancer. These are variants that are normal in the population. So we need to understand between the 30 or 40 million of variants that we have in the genome, which one are the important ones. So one approach is to study families with late onset Alzheimer's disease. I say that familial Alzheimer's disease is associated with mutation in APV, PS1, PS2, but this is not always the case. There are families that don't have mutation in these genes, and we think they have mutation in other genes. So what we did in my lab was to generate whole exome sequencing, and it was a pilot project, in 14 families, and for each family we sequenced several cases and several controls. We identified the coding variant because that's what you do when you do exomes, and by doing this, the next step was to identify the variants that were present on the cases and not on the controls within the sequence family. The next step was to identify which variants are rare, because if a variant is common, that variant is already identified by the GWAS study. And the end, we have around 75 non-coding variants for the segregated disease status within the sequence individuals. We have DNA for additional family members, we tag those, and we have only 8 variants per family. At this moment, what we did is similar in the representation has suggested, we see whether the same gene or the same variant show up in more than one family. And we were quite lucky. We identified one variant in one gene. It's called PLD3. That was everything disease status into family. So in the next study, what we did was to validate that this variant was real, and we genotyped this variant in more than 5,000 cases and 6,000 controls from different studies. As you can see in that forest plot, that variant was increased in 80 cases compared to controls in all the studies. And when you combine, you have a very strong ratio of 2 and a strong p-value of 10⁻⁵, which is quite good for rare variants. So these results already suggest that this gene is another gene for Alzheimer's disease. So when we identified this gene in 2014, we did a PubMed, and there were 3 papers about PLD3, nothing else. So this is a good thing, and the bad thing of genetics is that it's hypothesis-free. The question is, you don't know what you're going to identify, and you don't know what you can do in the next step. So obviously what we did was to analyze whether PLD3 is involved in APP metabolisms, because we know APP is a central pathway in Alzheimer's disease. So we overexpress and we knock down PLD3 in a cell system. And what we do consider is when we overexpress PLD3, A-beta 42 and 40 in extracellular levels decrease, and when we knock down, we see when we knock down A-beta 40 and 42 increase. Clearly, this data suggests indicate that PLD3 is involved in APP metabolism, but we don't know by which mechanism. Since the publication of this study, we have done additional analysis using classic biochemical studies, but also using RNA-seq from brain tissue for the PLD3 carriers. And we think that, or we are pretty sure, that PLD3 affects APP metabolism by affecting APP trafficking through the lysosome pathway. Okay. So this is some examples of how we can identify rare variants. There are more groups now doing this kind of studies. Now we have 300 families instead of 14. There is a very interest for the NIH to identify novel genes with sequencing studies, and they are also relating whole exome sequencing for 10,000 kids and controls. But as of today, this is what we know about the genetics of Alzheimer's disease. In this plot, we have the effect size of the genes, and you can see in the top APP, PS1, PS2, which cause Alzheimer's disease, but these genes, the mutation in the genes is very rare. On the other side, we have very common variants, but with very small effect size. And in the middle, we have ApoE and the novel genes that we have identified. We expect that in the next year, we are going to start filling this gap. But the interesting thing of this plot is that when we color code the gene by the function, we can see that some colors are more predominant than others. We obviously have the APP metabolism, but we also have a lot of genes that are associated with immune response. As I said, we still don't have any drug or target for to treat Alzheimer's disease that affect the immune or inflammation, but there are a lot of groups working on that now. So this is just my last slide. This is just a summary, and the summary is quite clear. There are 3 genes that cause the main form of the disease, and based on those genes, there are now several therapies that are on clinical trials. And these clinical trials is for all types of Alzheimer's disease, sporadic and familial Alzheimer's disease. Even mutations are only in the families. In the GWAS studies, we have identified the 20 risk log sites. The important thing of these genes is that they are part of a specific pathway, and those pathways are potentially a good therapy or target for therapy. Obviously, whole exome and whole genome studies are going to identify novel genes, and those novel genes hopefully will lead to novel therapies. But again, we are way behind the other fields like cancer or diabetes. And yes, I want to finish acknowledge all the people that have done the work in my lab, quite a lot, but also the funding agencies, which is basically the NIH, some pharma companies, and then private foundations. So thanks very much.
Te preguntamos.
Es muy interesante. Sí. Es increíble cómo el campo ha desarrollado este problema tan masivamente complejo de la última vez que yo lo miré hace 25 años.
Sí, los resultados básicamente son los últimos 7 años.
Mirando la población que tiene la variante más prevalente patogénica de APOE4, se podría mirar la presencia de alelos protectores. Ese es un alelo suficientemente frecuente en la población como para ahora introducir un estudio de protección de todos los individuos que tienen APOE4 risk factor,
Sí.
intentar ver cuáles no desarrollan Alzheimer y qué variantes genéticas pueden estar antagonizando el efecto de APOE4. Quizá esta sea una forma de mirar directamente a qué dianas terapéuticas pueden antagonizar ese pathway.
Sí. La frecuencia de APOE4 en la población es acerca del 20%. Aquí muchos en esta sala van a tener la de APOE4. En Alzheimer, en casos con Alzheimer es alrededor del 50%. Entonces, lo que dices tú, efectivamente es una muy buena estrategia y hay varios estudios que lo que se dedican es a comparar individuos con APOE4 en los extremos de la distribución. Son individuos con Alzheimer con early onset, comparando con APOE4, comparando con individuos que tienen 80 años o más, que son APOE4 y clínicamente están bien. Entonces, comparando eso, es posible. La idea es identificar variantes protectoras y ver si se identifican nuevas targets.
Hola, gracias, es interesante. Estaba pensando, hay una estimación, supongo que habrá gente que habrá hecho la estimación de cuál es la contribución genética al riesgo versus contribuciones de este tipo y cuánta de esa contribución genética, si se sabe o tienes buenas estimaciones, está explicada por las variantes que tiene. ¿Cuánto es lo que se espera descubrir en los próximos años si se llenan los gaps que estamos diciendo?
Los estudios para estimar la contribución genética son siempre complicados, pero hay estudios que dicen que es en late onset, o que si es familiar, el 100%, va entre el 50 y el 80. Aunque se llama late onset o esporádica Alzheimer's disease, en la mayoría de los casos siempre hay historia familiar de demencia. No es una historia familiar perfecta que es dominante, pero siempre hay mi tío, mi abuela tenía demencia. Entonces, es esporádico, pero siempre un poco de historia familiar. Entonces, es normalmente entre 50 y el 80. Rango y amplio. Los estudios de GWAS y las señales que se han identificado en los GWAS se cree que explican la mitad de lo que se ha estimado. Entonces, también por eso hay mucho interés en hacer exon chip y whole exon protein. O sea, creemos que no hemos encontrado la mitad del componente genético.
Y otra pregunta breve. En las noticias que estaba hablando hace un par de semanas durante la cena, me gustaría que lo discutieras en público, sobre que estos casos en los que se pasa a... que parece que hay transmisión porque recibieron determinadas hormonas de crecimiento.
Sí, la teoría dice básicamente una nueva teoría que Alzheimer's disease también puede ser una enfermedad como de prión, como las vacas locas. Entonces, lo que sí sabemos de hace no mucho tiempo, pero en los últimos 3 años o así, es que una vez que A, beta y tau se depositan en el cerebro, esos depósitos infectan a las células alrededor. Es decir, los depósitos no son aleatorios en el cerebro, sino que empiezan en un punto muy específico y se van esparciendo en el cerebro por las neuronas networks. O sea que en realidad hay un CNF. Entonces, lo que sabe también que en animales, si coges un ratón modelo Alzheimer's disease, le extraes y purificas a beta y se lo extraes y se lo inyectas a un animal wild type, no transgénico en el cerebro, ese animal va a tener placas. Entonces, había un estudio reciente que dice que también eso puede pasar en humanos, específicamente en individuos o en personas que han tenido o se han inyectado drogas para
multiplicarlo. La verdad es que nos hemos alargado media hora más. Yo creo que ha merecido muchísimo la pena. Nos ha encantado escuchar los últimos avances en la
ha sido una muy buena participación. Bueno, en general todos los ponentes, los cuales han estado orgullosos. Muchísimas gracias por vuestro tiempo y esperemos que abramos un poco más de debate ahora, bueno, después en la hora del café. Gracias por asistir y sigue creciendo.
Usamos